Hello, and thank you for watching this ChimerMarker webinar. My name is Kayla Hendricks, and I'm a biologist at Soft Genetics. Today I'm going to be talking about longitudinal monitoring using ChimerMarker software. Now, this is an advanced topic in ChimerMarker, so if you haven't watched the ChimerMarker introductory webinar yet, I encourage you to first go back and watch that webinar and then proceed with this webinar. To be specific, when I say longitudinal monitoring, I mean analyzing a chimerism project over an extended period of time. ChimerMarker facilitates this by allowing the user to create project files which can later be expanded with future data runs. In this webinar today, I'm going to show you exactly how to do this, and we're going to cover several aspects of creating chimerism projects and adding files to the projects. Just to summarize, we're going to be reviewing the basics of chimerism analysis. We will discuss how to generate longitudinal reports, which is a very useful reporting feature for longitudinal projects. This is an example of a simple longitudinal report showing the percent donor over a period of time. This next example is an in-depth example of a longitudinal report showing multiple cell types and the percent donor reported over a period of time for each cell type. We will discuss how to save project files. And finally, we will discuss how to add samples to an existing project, thereby expanding that project. So let's just jump into an example project. Okay, so here we are at the splash screen for ChimerMarker. To open data, we're going to click this icon here and then click Add. You can navigate to where you have your data files saved. Control A will select everything in the folder as a shortcut. And when you have your data files here, we'll click OK, and we're going to open up to our raw data. You can open multiple samples by just double-clicking on the sample name, and their electropherograms will open up in the main screen. You can scroll through the different dye channels using the Show Color icon and view all your raw data. So I imported here a ladder file, a recipient file, a donor file, and some post files. ChimerMarker automatically will recognize specific samples depending on the file designations that you have under the View Preferences. Along with the recipient and donor, you can also have different cell types to be auto-recognized. If you'd like to set up this, you can navigate to View, Preference, and the Chimerism tab. At the very bottom, there's going to be options for the donor, recipient, and cell type settings, and we can open this window and set in our designations for the recipient, donor, along with the cell types. In order to edit the cell types, you need to right-click, click Edit Cell Type, and a text file dialog will open where you can enter in your different cell types and click OK. Once you have these set, we'll click OK again, and your preferences are going to be applied. Once you're ready to go, we can analyze the data, and you can use the Run Project icon or Project Run. This is going to open up our Run Wizard, where we have some preloaded templates for different chemistries, and these samples were amplified with Promega's PowerPlex Fusion. So I'm going to select this template, and we have all of our panels and size standards preloaded for this kit. And as I'm using a ladder in with these files, I'm going to first create my calibrated genotyping panel. I'm going to use this panel to then create the chimerism panel so that all my peaks align correctly in their bins. On this last screen, I'm going to leave the auto panel adjustment option on in order to calibrate our panel to our ladder file. And we'll go ahead and process our samples. Done very quickly, and over to the left we can see that our ladder bolded, which is great. That means that our panel was able to calibrate successfully. And you'll just want to review the size calls in your donor recipient sample to make sure that everything is falling in their bins and meets your laboratory's SOP. Once you've done that, you can navigate to Tools, Panel Editor, where we can save our calibrated panel. And we know that this is the calibrated panel because it's going to list the ladder file after. So you can open File, Save as New Panel, and then enter in whatever you would like to designate this panel as your calibrated panel. And when you click OK, it's going to be added to our panel templates list, and we can access it from the Run Wizard. So let's go there now. When I reopen the Run Wizard, I can find my calibrated panel from the drop-down, and we'll go to Next. And this time we're going to go ahead and leave Auto Create Chimerism panel on, along with Auto Call alleles present in the chimerism panel. On the final screen, I'm going to turn the auto panel adjust off. This way no more shifting occurs because we have our calibrated panel already. Now we can see, as we look at the samples, that we have our donor recipient flags. And if you have any peaks that are flagged with a yellow or red allele label, you'll want to go ahead and just go through and make any edits that you need to according to your laboratory's SOP. Once you've done that, you can go to Applications, and we'll go to the single donor chimerism analysis. If you have a double donor project, you would just select double donor, but we have single donor, so we're going to go here. And I'm going to go ahead and use my default settings. And when we get into the chimerism analysis, you want to click on one of the mixture samples, and you can view your percent chimerism for each of your samples. All of this up until now should have been a review. So now I'm going to go ahead and show the new feature, the longitudinal report. Initially, the graph will be blank, so we'll need to go ahead and add some samples to it. Samples can be added using the checkboxes in front of the sample names, or you can select a sample with the left mouse key and drag to highlight other samples. Once you have the desired samples highlighted, you can right-click and choose Select, and this will also add the check marks to the boxes. Once you have the desired sample selected, you can use the refresh icon, and it's going to go ahead and add those samples to your chart. So you can initially see here that I have some different cell types graphed, but if you would have only a single cell type, your bars are just going to be the same color. So in the table below here is all of our information for our samples, and I'm going to go ahead and alter the date so we can see some of the monitoring over time. And once you have any changes that you make, you can then click the refresh button and it'll update your graph accordingly. So another feature of this table, along with being able to edit your date, and you can also edit the cell type, this is going to access those view preferences that we discussed previously in the webinar. You can sort these columns by clicking on the column head, and it's going to go ahead and sort them in terms of alphabetical order for the tissue origin, and then also the date is going to go in either the oldest to newest or newest to oldest, however you have it sorted. We can see that the table is initially, it's going to be a bar chart, but if you'd like to see a line chart, you can go to the graph display, choose line chart. It's going to update your graph accordingly. So I have Show 3D on right now, but if I turn it off, it's going to take us back to a 2D view. And the Show 3D is nice when you have the bars. It gives you a little extra depth to what you're looking at here. You can also change display settings using the display settings icon. You can change the settings for your labels, the date format, along with the chart settings, how many layers you'd like to display, and also some picture saving options. And once you have that all set, we can preview our actual report, and that can be opened using this print icon and preview. And it'll give you an example of the report that can be either saved or printed. So the initial page will be your graph, along with the second page containing the table, and the third page for any comments that you have. And you can also notice that there is a header. The header is going to contain the version of ChimerMarker you're using. If you have user management turned on with your institution's name here, this will be displayed instead of Soft Genetics. Your project name will show up along with the user who is currently logged into user management. Also, the analysis type, the panel, and the chimerism type and your quantification method will all be in this header at the top here. And if you look on the left, we have some recipient information that is not filled in at the moment. So we can go ahead and fill that in now, and I can show you how that's going to display on the report. So the recipient information can be added in the longitudinal report or back in the chimerism analysis window. So you can add in a case number, a name, along with gender, birth date. If you have a date, you can just enter the date and your age will be calculated automatically. And you also have your transplant information, and you can enter in a donor, some donor information. along with the comments, and these comments will be displayed on the last page. When you click OK, this information is going to be updated in the top of the longitudinal window here, and if we go back to the report, you can now see that our header is going to contain all that information that we just added. After you have all that information and you've looked at your time points, you can print this graph or save it, and you can also go back to the main analysis screen and we'll save our project. So from the file drop down, we'll do save project, and you can name this with today's date or whatever you would like. And click save, and now our project's saved. So we can go and we can close out a ChimerMarker. Some time has passed and we have future data runs that we'd like to add to this longitudinal report. Instead of having to reanalyze all of our samples from the beginning, we can go to File, Reopen Project, and open that project that we previously saved. And what you can see here is that all of our information that we added is saved. Our chimerism analysis settings are going to be saved. If we come back into the mixture, any edits that were made will be saved in the project along with recipient information here, and our longitudinal report will be as we left it also. So with all that saved, since we don't have to reanalyze, we can add samples to our project. So this can be done going to the project drop-down, clicking Add Samples to Project, and then we're going to get an Open Data File screen like we've seen before. So then you can go ahead and navigate to where you have your new post samples, select all of them, and we will bring them into the program. And while these are processing, it's going to go ahead and use all the template and the Run Wizard settings that you have already set. So all your samples are going to be analyzed the same way. And so now that they've been added, we can go back into the single donor report. We'll use all the same settings, and now we have our additional samples available here. And when we come to the longitudinal report, we can see that our new samples were added to the bottom. So in order to add them to the graph, it's just going to be the click and drag again and select. I'm going to go ahead and give us some future dates. So again, we can see more of our longitudinal reporting. And now that we have all the dates set, we can click the refresh icon and our graph is going to be updated. And so again, you can go ahead and you can print and save your additional graphs here. And once you have all this set, you can then save your project again, and you can choose to save the same project and replace that, or you can add a new name so you don't overwrite that existing project. So those are the basics of longitudinal monitoring in ChimerMarker. One final thing I wanted to mention is many groups that are switching to ChimerMarker have longitudinal projects that they already have started and are ongoing that may be quite old. In some of those projects, it is possible that the original FSA or .HID files are no longer available for the donor and recipient samples. We often get the question, can those longitudinal projects still be analyzed in ChimerMarker even if the donor and recipient files aren't available? And the answer to that question is yes. If you have the donor and recipient genotype saved in an Excel file or in a text file of some kind, we do actually have a tool called the Text Import tool, which is on the second page of the Run Wizard. It allows you to upload the genotypes of the donor and recipient files in the form of a text file. So those projects can actually be accommodated in ChimerMarker, and you can analyze them fully again if you have the genotype saved, but don't necessarily have them in the .FSA or .HID files. And please view the separate webinar for more information about the Text Import tool. If you have any other questions, please just stick around to find out how you can get more information. Thank you for watching this ChimerMarker webinar. For more information or for a free 35-day trial of ChimerMarker and other SoftGenetics products, please visit our website at softgenetics.com or send an email to info@softgenetics.com. Of course, you can always send any technical support questions to tech_support@softgenetics.com. Thanks again for watching.